Hi learners, welcome to the 20th and the last lesson of my course Ecology from Cells to Gaia. Today we will talk about you as an ecologist. In recent years, surveys and studies on biodiversity have become so important that more and more civil society representatives join research and propose partnership with research institutions that are working in the field. From this growing interest, a new survey technique was born, which is called BioBlitz. This is an intensive biological investigation which aims to record all the species living within a designated area, comprising a group of specialists supported by no experts. Scientists, naturalists, and volunteers carry out an intensive field surveys of short-term, usually 24 or 48 hours. Even if you are not an ecologist, you can organize your own BioBlitz. In this lecture, I will show you how to do it. The public component of the BioBlitz is of fundamental importance in spite of the lack of naturalistic specialization of many participants, and is able to provide an unimaginable quantity of data on the biological diversity. Usually qualified biologists and naturalists are able to train every involved citizen in identifying species in the field. Generally, a BioBlitz proceeds as follows. First, sites of interest for the study and protection are identified from satellite and then in the field. Field surveys of biodiversity, geomorphology, ecosystem and anthropogenic impacts are conducted. Then data are collected and samples are catalogued. Finally, the results are analyzed. Various instruments are used in the field, such as binoculars, portable microscopes, tweezers, transparent bags, notebooks, cameras, and others. To conduct more accurate BioBlitz, 3 research campaigns for each selected site can be carried out at about every 4 months in order to detect changes of environment and the variation of biodiversity during each season throughout the course of the year. The preparation of a standardized protocol allows to conduct field operation in a coordinated manner and can be organized following this example. First, during the field campaigns, it is necessary to take notes on the notebooks given to each participant. The identification number of the sampling area of the plot or the sample should be written, the name of the place, the date, name of the researcher, the weather condition, and all the species identified by direct or indirect observation should be reported. It is important to indicate at least the common name, if applicable, and the scientific name of the rare species identified in each group, mammals, reptiles, amphibians, etc. When in doubt, it is better to indicate the genus or the family and to specify the feature to later recognize the species. For a later identification, it is better to collect leaves, fruits, droppings, or pellets. Therefore, bags for the samples collection and adhesive labels on which to indicate the serial number, the sample length and the shape, the place of discovery, for instance on a stone, into the soil, in a latrine, the color, the smell, the remains presence from plants and animals, and if possible a reference to family, the genus, and the species to which the sample may belong are made available too. An identification number and the first 3 letters of the surveyor name must be assigned. For all the samples, it is necessary to take a photo before the collection with a reference object, a coin, a lidar, placed in the proximity. For plant samples collected, that is for those plant species not directly identified in the field, the same steps listed in section 2 must be performed, while for samples not collected, for instance those easily identified, it is sufficient to take note of the species name on the datasheet and in case of doubtful taxonomy, just to take a photograph. For each sample collected, a reference number must be associated in the datasheet. On the back of the datasheet, observation on the environment, possible threats, geomorphology, etc. can be annotated. Since activities involving citizens are likely to disturb local ecosystem involved by BioBlitz, a regulation should be made on the basis of the following recommendations. First, it is necessary to ensure silence and attention during the field campaign. So if there is a need to work in a group, it would be desirable that these do not exceed 3 components. 2. In order to don't collect a necessary surplus of samples, it is important to make use before sampling of the opinion of other members of the group that could identify the species, preventing the withdrawal. 3. For the purposes of research, live specimens are usually not necessary, therefore it should be absolutely forbidden to sample live animals or kill them, even if are insects, but only identify and photograph them. The skeletons or dead animals are, however, to be taken by the same procedures described above. 4. While walking, it is necessary to create the least damage to the flora, by no withdrawal more than necessary plant parts for identification and never collect orchids, and the least disturbance to wildlife. 5. Because the project is carried out under the legal and moral responsibility of association entities university, people that do not respect the agreements made and the information provided will be advised to not continue the field campaigns. Each surveyor involved in the field campaign should be equipped with the proper clothing and instruments. For instance, camouflage clothing, green or brown, comfortable shoes, hat and raincoat, enough water and picnic lunch, backpack, binoculars of different magnification, digital cameras, flexible centimeter or calipers, electronic pocket scale, notebook, pen and pencil, datasheet for surveying, transparent plastic bags for the collection of samples, adhesive labels, naturalist pocket guides, compass or GPS, medicine for the first aid, and maps of the study area. The daytime field research phase should start preferably in the morning just after the sunrise and end in the midday or evening depending on the time required to monitoring the study area. After a survey of the field instrumentation of each participant, it is important to locate the sampling transect so individual researchers or groups of 2-3 people can keep a distance of at least 5–10 meters from each other and ensure coverage of the entire area, moving in lines parallel to the transect as a rake. At the end of the field campaign, all samples and all datasheets should be collected by the responsible person for the subsequent phase of analysis in the laboratory. In addition to daytime research campaigns, night field surveys can be carried out, which include camouflage in sighting huts and diets to study animals with crepuscular habits, for instance with infrared night visor or camera traps. The research on bird populations can be conducted by means of direct sightings, use of hides, photo traps, and indirect observation, eggs, feeders, pellets. Entomological studies can be realized by means of entomological umbrella method, which consists of opening a towel at the bottom of a tree and the shaking of it to bring insect down, or indirect or direct determination. For reptiles and amphibians, direct identification and photographic techniques are generally used. Mammals, however, may require direct identification day and night in dark reliefs, nests, burrows, airs, footprint, excrements, camera trapping, and laboratory analysis, for instance for remainings of small mammals in pellets and faces, determination of teeth formula, and skulls. Plant species can be determined instead by satellite analysis, direct identification, sample collection, and laboratory analysis. The bioblitz in rivers and lakes, as well as those in marine environment, require the presence of experienced staff and higher security measures. After each campaign, all data collected and all samples must be transferred in the laboratory, catalogued and analyzed. In addition, all information in the datasheet should be converted into electronic spreadsheet format to allow the subsequent creation of an electronic database. Each datasheet must be subjected to cross-check in order to verify the correctness of the taxonomic nomenclature and the matching of the species detected with the bibliographical information available and with the species distribution ranges. Uncertain and undetermined species can be classified by means of guides and consulting several experts. Animal and plant samples should be subject to preliminary analysis by naked eyes and then be analyzed with a stereo microscope. During laboratory phases, photographs taken throughout the field campaign can also be analyzed and catalogued, and a photo database can be created with area and species information. The BioBlitz could be a very interesting experience for you after following this course because you will apply all the information provided in this course and during the lecture. So please, I advise you to follow the instruction provided and to create your own BioBlitz. So I hope that this course taught you how wonderful is our planet and why we should protect it. So please take care of it.